This protocol allows plant-wide real-time imaging of the activity of the plant systemic signaling system through monitoring the dynamics of the calcium and apoplastic glutamate in response to wounding. This plant-wide real-time imaging method provides a robust tool to understand the dynamics of the rapid and long-distance signals in plants, combining a high spatial temporal resolution and ease of use. This protocol offers the potential to provide a new insight into the spatial and temporal characteristics of the systemic calcium signaling in both biotic and abiotic stress responses in other plant species. Demonstrating the procedure will be Takuya Uemura, a postdoctoral fellow from my laboratory.
Begin by turning on the motorized fluorescent stereo microscope equipped with a 1x objective lens and a SCMOS camera. Configure the device settings to irradiate with excitation light centered on 470 nanometers, selected using a filter that transmits light between 450 and 490 nanometers. Acquire emission light using a filter that transmits between 510 and 560 nanometers. Remove the lid from the dish containing the plant and place it under the objective lens. Check the fluorescence signal from the plant. Then wait for approximately 30 minutes in the dark until the plants are adapted to the new environmental conditions. Adjust the focus and magnification to see the whole plant in the field of view. Then set the acquisition parameters to detect the fluorescence signals using the microscope's imaging software. Set the recording time to 11 minutes. Image for 5 minutes prior to starting the experiment.